Hello, everybody, and welcome back to BioSC 140 Human Physiology. This video is going to cover S3P3, the cardiovascular system. This is lecture exam 3 material. So we're going to start off talking about the vessels. So cardiovascular system. What are the three main components of the cardiovascular system? We've already talked about two. So we have the cardia, which refers to the heart, and then the vascular, part of the word is the vessels, the tubes, and then it's the stuff that's inside the tubes, blood. So we've already covered blood and the heart. Now we're going to cover the tubes. So vessels are conduits that move blood from place to place. They are the tubes that blood moves through. There are differences in the structure, in the form of the different tubes in our circulatory system. Remember, circulatory system is the functional name of our cardiovascular system, more of an anatomical name. So you should know the general flow of our bloodstream, of our circulatory system. So we have our left heart, which generates pressure and moves blood into our aorta and our arteries. That'll carry blood to arterioles, which will carry blood to capillaries, which are the sites of gas and nutrient exchange, which will carry blood into our venules, which will carry blood into our veins, which will carry blood into our right heart, which will carry blood into our pulmonary arteries, which will carry blood into our pulmonary capillaries, which will carry blood into our pulmonary veins and back to our left heart. So let's talk about arteries first. So arteries take blood away from our heart. Away from our heart. Sometimes people get a little mixed up and they say arteries only carry oxygen-rich blood. There's an exception, the umbilical and pulmonary arteries. So if we come back here, arteries in our systemic circulatory system carry oxygen-rich blood right here. Arteries in our pulmonary circulatory system do not. So I'm probably going to ask a test question on that, and any class in the future on the cardiovascular system, your teacher's probably going to ask a question around knowing that knowledge. It's easy to write questions on, and it always mixes students up. So be ready. I'm warning you, giving you the heads up. All right, so arteries carry blood away from the heart. Now, they have special characteristics that help them do this. Let's just get into writing. So they're muscular and they're elastic. They're muscular and they're elastic. There's smooth muscle in the walls of arteries, and there's also a high level of a lot of elastic fibers. So let's think about pressure. Remember, blood moves from high pressure to low pressure. Arteries need to maintain higher pressure than veins. And it's this elastic characteristic that allows them to constantly maintain a higher pressure than the capillaries and the veins downstream. So our left ventricle, when it's filling with blood, zero pressure. When our left ventricle is, at maximum contraction, maximum systole, pushing blood out, gets up to 120, pressure. Left ventricle is going to fluctuate between roughly 0 and 120 pressure. The aorta is going to fluctuate between 120 and 80, our systolic and diastolic blood pressures. The heart is going to pump blood in the aorta, and it's going to blow it up like a balloon. When the heart starts to relax, the semilunar valve is going to close, preventing blood from flowing back into the heart. And that expanded balloon is going to, with that expanded elastic properties, kind of like a balloon, is going to slowly squeeze down on that blood and maintain that pressure, maintain that slow pressure squeezing down on the blood until the next systole cycle comes from the left ventricle, which pumps it back up again. Arteries are like balloons. You pump them up, they maintain that pressure as they squeeze down. This helps maintain high blood pressure in arteries. Remember, blood flows from high pressure to low pressure. You need to maintain higher pressure in your arteries than in your capillaries and then in your veins. This is surprising to most students. A relatively low amount of volume of blood is found in your arteries. Only about 10% of your total blood is found in arteries. A small, 10%. So not too much. Right here, this is what I was talking about. The elasticity of arteries preserves pressure during diastole. So the heart pushes blood and fills up that aortic balloon. Systolic pressure is stored in the elastic walls for the arteries and slowly released through elastic recoil. That pressure that's built up is slowly released through elastic recoil. This maintains the driving pressure during relaxation. This helps maintain that high pressure, that inflated balloon pressure in our arteries, so that there's always higher pressure in arteries than in capillaries and veins. You can see right here, semilunar valve closes, and that pressure wave, that elastic characteristic, maintains that push forward for blood. Arterioles. Arterioles are thinner than arteries. They're more narrow than arteries, and they don't have so much of an elastic characteristic because their job isn't so much to maintain that driving pressure, that driving blood pressure. Arterioles, their job is to partition blood flow. It's to decide where blood flows to. And so they are very muscular. Arterioles, if you remember back to our first class, how I talked about how the rabbit can send more blood to its ears or less blood to its ears, it's arterioles that decide where that blood is flowing to. When you put your hand in ice water, if we did the ice water lab and the senses lab in real person, you'd see this, or just on a cold day, it's cold now. You go outside without gloves on and your hand, might get a little pale when it gets cold. Your body will automatically send less blood to your hands because it doesn't want to lose that heat. And what it does is it constricts the arterioles. So the arterioles constrict, you have less blood flowing into that area. Let's say you start doing a bunch of bicep curls. You need more nutrients to go to your biceps. You need to get that CO2 out of the area, all those waste products out of the area. Your arterioles will dilate in the biceps that you're working out, allowing more blood to flow to the area. Arterioles have a small total cross-sectional area, and they're the greatest site of vascular resistance. The greatest site of vascular resistance. The diameter is highly regulated by vasomotor tone. So vaso meaning vessel, motor like muscle, tone like there's a level of contraction, and it can relax or contract all the time. Right here, put some stars around this. Put some stars around this right here. Sympathetic innervation only. Arterioles are innervated by the sympathetic nervous system only, not the parasympathetic. They're also affected by hormones. So epinephrine can cause constriction. Epinephrine causes constriction of arterioles. Local chemicals. So local chemicals are going to be, they're actually usually byproducts of metabolism. So carbon dioxide, lactic acid, nitrous oxide. Local chemicals, when they're usually waste product chemicals, When there's more of them around, arterioles dilate. Local chemicals cause dilation. Those local chemicals are usually waste product chemicals that, when created, um, cause arterioles to expand so that there's more blood flow, there's more of an ability to get those waste products out of the area. So these arterioles are gonna, arterioles direct the distribution of blood flow. So this is what I was talking about with, you work out your bicep, more blood flows to the area. That's due to production of local chemicals, which dilates those arterioles. It's cold outside. Your hands get cold and start losing a lot of heat. So your body wants to restrict blood flow to your hands. Sympathetic nervous system tells your arterioles to send less blood to your hand. You get less blood flowing there. Capillaries are the sites of cellular exchange. So capillaries, their job is to exchange. They're like the bus stop. They're the train station of your circulatory system where things get on and off. So remember back to Fick's law, test 1. The thinner the wall of the membrane, the more flux there is. So capillaries are very thin walled. They have simple squamous epithelium. It's a single thin layer, very thin layer of endothelium. And this helps the function. So form and function, we're seeing it again even on test 4 material. It allows fluid to move from the plasma, the blood plasma, to the interstitial fluid, and stuff to move from the interstitial fluid into plasma. The junctions between cells allow for variable leakiness. So it's those spaces between the cells that allow for stuff to kind of ooze in and ooze out. And different, there's variable leakiness depending on what's going on in the area. White blood cells, if white blood cells need to get to the area outside the capillary, they can actually make the capillaries more leaky, for instance. So there's a number of things that can affect the leakiness. Capillaries are narrow. Capillaries are narrow but numerous. So in the cardiovascular lab, normally we look at a fish tail underneath a microscope, and you can actually see how capillaries in that fish tail are one cell wide. You see one red cell kind of just squeeze through that capillary and shoot through that capillary at a time. Capillaries, only one red cell can squeeze through those capillaries at a time. But they're numerous, around 50,000 miles of them in a human. That's insane. That's a lot of capillaries. So they have the largest cross-sectional area of all vessels. Largest cross-sectional area of all vessels. And this actually slows the velocity of blood. So when blood gets to capillaries, because of this wide cross-sectional area, it actually slows down the speed of the blood, which allows for more time for diffusion, more time for things to move in and out. Form and function, once again. So capillaries are the sites of cellular exchange, and whether things move in and out of the capillaries, it's dictated by the Starling's forces. Starling's forces, there's the hydrostatic pressure within the capillary, which is related to MAP, mean arterial blood pressure. That's going to push fluid out. And then we also have the that colloid osmotic pressure of the interstitial fluid, like pulling material out of the capillaries. Then we have the hydrostatic pressure of the interstitial fluid, pushing fluid back into the capillary. And we also have the osmotic pressure of the blood, pulling things back into the bloodstream. So there's two osmotic forces inside and out, pulling fluid towards those dissolved particles. And there's the hydrostatic pressure within the capillary and outside in the interstitial fluid, pushing fluid in the opposite directions. On the arterial side, it tends to have net filtration, so things flowing out of the capillary. And on the venous side, it tends to have net reabsorption, so things flowing back into the capillary. Veins and venules act as a volume reservoir. So most of your blood is stored in veins, actually. Veins have a high capacitance. They're floppy. They fill easily, and they have valves which ensure one-way flow. So veins are low pressure. Veins are low pressure. They need to maintain low pressure because things, you want it to flow from arteries to arterioles to capillaries to veins and venules. They're like they don't have much of a stretchy characteristic, a little bit of a stretchy characteristic, but not much. They're collapsible. That's a good way to put it. They can kind of go flat and expand to a tube, then go flat or be somewhere in the middle of those. They're kind of floppy. They fill easily. They have valves which ensure one-way flow. We're going to talk about this soon. This actually has some healthcare implications. Because they're so low pressure, the pressure gradients aren't always set up for blood to flow in the direction that we want. And so there's valves and veins which allow blood to flow towards the heart, but prevent blood from flowing away from the heart. Function: return blood to the heart, dissipate heat, and store blood. Most blood is in veins. About 60% of our blood is in veins. Veins can constrict and they can dilate. When veins constrict, it increases venous return, which means that when veins constrict, they help push blood back to the heart. They increase their pressure, still remains low pressure in the atria and the ventricles at the right time of the cycle, at least. So you constrict the veins, increases the pressure, and it pushes that blood back towards the heart. It can't flow the wrong way because of the valves. Then dilation will increase blood storage and decrease venous return. So right here, we can see another thing that's kind of interesting about veins. So we have a valve here, and we have a valve here. Pressure gradients help drive blood back to our heart. And sometimes that pressure is actually generated by our muscles. So right here, valves in the veins prevent backflow of blood. When skeletal muscles compress the veins, they force blood back towards the heart, the skeletal muscle pump. You can see right here, the calf muscles are constricting and putting pressure on the veins, which is going to increase the pressure. It can't flow backwards because of the valves, but it can flow back towards the heart. So skeletal muscle can help the blood flow back towards the heart, venous return. Right here is a summary chart of all of it. Whenever you see a summary chart, you know that information is important and you should know pretty much everything on it. And here is another summary chart, 2 summary charts, twice as very important stuff. Definitely study these summary charts. Watch this video a few times. Let me know if you have any questions. There will be a good number of questions on this material. Well, let's go on for a few more slides while I was talking about veins. So a portal system. So up until this point, in the flow of blood down here, there's been one capillary bed every time. That's the rule, except for in 3 locations. There are 3 locations where there are 2 separate capillary beds in line, in series. So portal systems is what they're called. Portal systems have 2 capillary beds in series. You should know them. The hepatic portal vein right here. So blood flows, it goes to the digestive tract, it picks up water-soluble nutrients, and then it goes directly to the liver where there's another capillary bed. So capillary bed in digestive tract, capillary bed in the liver, and then blood goes back to the heart. There is the hepatic artery, which goes straight to the liver, but we also have this one right here, the hepatic portal vein, is that vein right in between the digestive tract and the liver. So we have O2 delivered by the hepatic artery right here, and then we have nutrients that go right when we absorb them. We want them to go to the liver for processing. They go straight to the liver for processing. That's the importance. Absorb nutrients, straight to the liver for processing. We have the hypothalamus and pituitary system. So up in our brain up here, we have one capillary bed in the hypothalamus, and then we have a vein, and then another capillary bed in the pituitary gland. The hypothalamus will actually release hormones into that capillary system, which goes right down right next door to the capillary bed in the pituitary gland, which stimulates the pituitary gland to make more hormones and do various things. Then we have the capillary bed in our kidneys. So we're going to cover this in lab and in lecture. This form and really affects its function. So we're really going to dive into this a lot in lab and lecture. Not this lecture, but part 4 lecture. And we'll see it in lab 27. So we have one capillary bed called the glomerulus, or the glomerular capillary right here, which is kind of the entrance to the nephron, which is the functional unit of the kidney. Then we have our afferent arteriole, and then we have our peritubular capillary, which is another site of exchange. And then it goes to the renal veins. So portal systems have 2 capillary beds in series. You should be familiar with all 3. And with that, I'm going to conclude part 1. As always, if you have any questions, please let me know. I'm here to help. See you guys in the next video.